Today, we're talking all about cellular respiration. We've been building up to this. This is has the reputation of being one of the harder concepts in the course. I don't think it's okay. I don't think it is. I think we can break it down to the point where it's just 4 steps. As long as you got those 4 steps, you got cellular respiration. The reason we're studying cellular respiration is because it gives us a model. Just like photosynthesis, it gives us a model for how metabolic pathways work. And the reason we study these pathways is so that you guys get an idea of sort of how science works systematically, how you can systematically problem solve and figure out how to go step by step through an issue. So, cellular respiration is where we're going to take energy from food molecules, we're going to get energy from food, and release waste products. That's really it. You get food from your environment because you're a heterotroph, you break it down. All right, what I want to point down, you're getting energy from your waste products, right? You're stealing electrons from these food products. You're stealing electrons from what you eat. So what are you doing to the things you're eating if you're stealing electrons? If those things are losing electrons, they're being oxidized. So you are oxidizing substrates. At its heart, all reactions that give you energy are going to be reactions where you oxidize the substrate. This is just one example. Most cellular respiration is motivated by the cell breaking down the bonds of glucose. Now, it doesn't just have to be glucose, it could be any bonds. But you get your energy from breaking bonds, from stealing electrons. Glucose is just one of the many, many substances we break down. I'm emphasizing this idea that glucose is just one of many things because after 101, people come out saying, well, you get all of your energy from glucose. You don't. You don't just go home and eat pixie sticks all day. Oh, you might. I don't know. But you can't man cannot survive on pixie sticks alone. You need more than just sugar. Ultimately, what you're doing is you're taking that food that you eat, you break the bonds, you steal the electrons, you oxidize the substrates. But for today, we're going to talk specifically about breaking down glucose. So, we get glucose in our cells, but where does it come from? Where do we get glucose? Yeah, food, right? And where does the food get its glucose? Where do plants get their glucose from? Light. So these autotrophs are going to produce glucose using photosynthesis. We, being heterotrophs, go and eat those autotrophs, and we can steal glucose from them because we're bigger and badder than they are. So we can break down glucose in one of 2 ways. We can do it with oxygen or without oxygen. When we're doing it with oxygen, it's called aerobic. And without oxygen, it's anaerobic. Think when it's you're doing aerobic exercises, you're breathing a lot, you're taking in a lot of oxygen. Anaerobic, you're doing it without oxygen. An means without. Today, we're concentrating solely on aerobic respiration, using oxygen to help break down glucose. So here's the formula for aerobic cellular respiration. You've got glucose, C6H12O6 plus 6O2 yields 6CO2 plus 6H2O. So you're taking glucose and oxygen, and you're breaking it down into carbon dioxide and water. As we break down glucose, as we break the bonds of glucose, we steal those electrons, those energetic electrons, and we can use those energetic electrons to produce this molecule. What is this molecule? That's right, adenosine triphosphate. And you can tell because it's got those 3 phosphates on it. ATP. So does that equation look familiar to you? Have you seen it before? It's the exact opposite of photosynthesis. If you look at photosynthesis, it's carbon dioxide and water, yields glucose and oxygen. Cellular respiration and photosynthesis are linked together. Now, where does photosynthesis occur? What organelle? The chloroplast. And what organelle does aerobic cellular respiration occur in? The mitochondria. Plants have both chloroplasts and mitochondria. They both create glucose, and then it moves over to the mitochondria where it can be broken down into usable cellular energy. We only have mitochondria. We have to take in our glucose from the environment, but then we break it down using this aerobic cellular respiration. There are 4 steps to this process. You get these 4 steps, you are set and golden. Glycolysis, the breakdown of pyruvate, the citric acid cycle, otherwise known as the Krebs cycle, and oxidative phosphorylation. Let's take that over. So glycolysis, breakdown of pyruvate, citric acid cycle or the Krebs cycle, and oxidative phosphorylation. The first step, glycolysis. Step number 1, we'll draw it over here. Glycolysis. So, glycolysis is the cutting of glucose, breaking down glucose, breaking apart. So, glyco, think glucose, lysis is to break down. So you're breaking down glucose. Anytime you break a bond, what happens? You release energy. So this whole process is going to be releasing energy from glucose. So this slide is wrong. It says it's common to all organisms in the world. It's not. I lied. This is to separate the people who come to class from the people who don't. Glycolysis is not common to all organisms. Some organisms will never find sugar in their entire life. Some organisms live on the bottom of the ocean, near geothermal vents. Light has never gotten down there. Sugar doesn't drift down on its own. Instead, they're getting their nutrients from breaking down rocks and breaking down material that's spewing up from the Earth's core. Glycolysis is common among living organisms, especially those on the surface. On the surface, we take in a lot of sugar, but other places they don't. It is a common process, but it is not common to everything. Glycolysis happens within the cytosol of the cell. So in the cytoplasm, between the spaces in between the organelles. Heck, you don't even need organelles for glycolysis to happen. It happens in prokaryotes all the time. So here's the basic pathway of glycolysis. I'm not going to ask you to memorize the whole thing, but I do want to go through it so you see what I'm talking about. You start off with glucose. ATP comes in and ADP is removed. So when ATP comes in and split, what's released? ADP. And what else? A phosphorus. Okay. A little bit more. Whenever you break a bond, you release energy, right? You have to add a little bit of energy to that glucose to get it ready for the rest of the breakdown process. So you turn glucose into this fructose diphosphate. You've added some energy to it. But then you split that fructose diphosphate. When you break it, what happens? Energy is released, right? So in this case, you split that fructose diphosphate into 2 molecules that release energy. One is in the form of NADH. One's in the form of ATP. So, and that produces what's called pyruvic acid or pyruvate. So 2 ATP come in. 4 ATP come out, giving you a net total of how many ATP? Well, 4 wow, that's a lot of different numbers. 2 go in, okay? You lose 2, minus 2. 4 come out, plus 4. Gives you a total of 2 ATP. I'll draw it out even more. Sorry, having a little bit of issues with that, I see. All right, so if we have, here's our glucose. And we need to start this reaction off. So we put in 2 ATP come in. Does that make sense? 2 ATP are coming in. Glucose gets broken. That means it gets built into fructose diphosphate. That fructose diphosphate's then broken into pGal. And in the process, 4 ATP come out. 2 went in, 4 come out, giving you a total that you produced in this whole process of how many? 2. Do you see that? Do I still lost you on that? Okay. Check this out. I brought string from my other class. Turns out it's going to come in handy here too. So that's glycolysis. You have 2 ATP go in, 4 come out, giving you a net total of 2. You have 2 NADHs produced, another energy intermediary. And you've gone from glucose, 1 molecule of glucose, to 2 molecules of pyruvate. Step by step, that glucose is going to produce 2 NADHs. NAD plus gets converted to NADH. That's going to come into play in the next lecture, so don't forget that. 2 NAD plus get converted to 2 NADH, an intermediary. In this process, you're going to produce ATP through substrate level phosphorylation. What is substrate level phosphorylation? Even better, what is phosphorylation?
Taking a phosphate from another molecule.
Yeah, pretty much phosphorylation is adding a phosphate. Now, substrate level phosphorylation, when you think substrate, what do you think of? Enzymes. So substrate level phosphorylation is using an enzyme to take a phosphate and add it to an ADP. You're phosphorylating using an enzyme. So glycolysis is substrate level phosphorylation. Tricky question, what's the other way that we know how to phosphorylate something? A very efficient way. Chemiosmosis. We're going to see that in a few minutes. But here we've made some super quick energy using substrate level phosphorylation. This glycolysis is also going to take a 6-carbon molecule and cut it in half. You take a 6-carbon molecule and cut it in half, what do you have left? 2 3-carbon molecules. And that line, it looks sort of like a balloon animal, right? A little dog. But you got 1, 2, 3 carbons. You cut a bond, you released energy in the form of NADH and ATP. And you have this 3-carbon molecule. So that's glycolysis. Let me write that up on the board. By the way, the best way, absolute best way to learn this is just like photosynthesis. Draw it, draw it, draw it. So we have glucose gets broken down into 2 pyruvate. 2 NAD plus go in, 2 NADH come out. You also have 2 ADP go in net total, and 2 ATP come out. This process is step 1, glycolysis. Questions? Get into step 2. We're going to have these 2 molecules of pyruvate are going to enter into the mitochondria. This is where we're going to have the breakdown of pyruvate. That means we're going to, go figure, break the bonds in pyruvate. When we break the bonds in pyruvate, what are we going to release? Energy, because when in doubt, that's what we're doing here. Now, this step does not happen in prokaryotic cells or archaeans because they do not have any mitochondria. So, no mitochondria, you can't have this next step. This is only going to happen in eukaryotic cells. So here's what we're going to do. We take that pyruvate. We're going to hack off carbon dioxide using an enzyme called pyruvate decarboxylase. I don't need you to know pyruvate decarboxylase, but just take a look at it. It's got the word pyruvate, D. What does D mean? If you're going to deworm something, what are you doing? Get rid of, right? So we're removing carbon and oxygen. So pyruvate decarboxylase removes carbon dioxide from the pyruvate. So we're going to remove CO2, leaving us with this molecule called acetaldehyde. Acetaldehyde. So we get 2 high-energy electrons removed from pyruvate, and that makes more NADH. The remaining piece, that acetyl group, is attached to coenzyme A to produce a molecule called acetyl coenzyme A. Let me draw that out. So we got 2 molecules of pyruvate. We're going to cut off, there are 2 pyruvates, right? So we cut off a carbon dioxide off of each one, giving us how many carbon dioxide released? Not trying to trick you. 2, right? 2 molecules of pyruvate, we cut 1 carbon dioxide off of each, so we release 2 CO2. And we also release NADH. In this case, we've cut that CO2 off, released some energy, 1 NADH per, so we have 2 NADH released. And that produces a molecule called, ultimately, acetyl combined with coenzyme A. So acetyl-CoA. We've seen coenzyme A before. Where have we seen coenzyme A before? Say it again. It attaches to something, but not to an enzyme. Okay, that would be the other thing it would attach to. Yes, it attaches to some substrate, but where and when did we see this? In the cell. What part of the cell? Narrowing it down. It's okay, you got that part. Somebody else will pick up the next. Where in the cell? If it helps, I specifically told you coenzyme A is going to come up again. And you know, actually what I told you was coenzyme A is going to come up again. And I will walk out of the room and get a drink while you figure it out. So the smooth endoplasmic reticulum is where we've seen this before. The coenzyme A was previously used to attach the phospholipids, attach the fatty acid tails to phospholipids. But here we're using it in a different context. Here we're attaching an acetyl group to this coenzyme group. Here is where we saw it originally, attaching it to phospholipids. Now we have a grand total of 2 acetyl coenzyme A's produced, 2 carbon dioxide produced, and 2 NADH are produced from the breakdown of pyruvate. That gives us step 2, the breakdown of pyruvate. Two more steps to go, and then we're done. Next, we enter into what's called the citric acid cycle, or the Krebs cycle, named after Hans Krebs. So what's going to happen here is that acetyl coenzyme A enters into a cycle. It goes in a big circle. Acetyl coenzyme A produces citrate to acnonitrate to isocitrate to alpha-ketoglutarate to succinate to fumarate to malate to oxaloacetate back to acetyl coenzyme A. I'm not interested in you knowing those steps, nor am I interested in you knowing the enzymes that produce them. What you need to understand, the big thing you need to know here, is that there are lots of steps. Each of those steps is an enzyme-mediated reaction. Now, if it's enzyme-mediated, that means you could speed it up or slow it down by environmental controls. If it's too hot, things start to, if it gets too hot beyond the optimal conditions, this process slows down. If you were to be in a high pH or low pH situation, this process would start falling apart. Does that make sense? Anything that impacts enzymatic reaction rates impacts this cycle. Oh, similarly, what else would impact it is inhibitors. Inhibitors are a big problem with this. Now, this was discovered by a gentleman named Hans Krebs. And Hans Krebs sort of, it's unfortunate, but he was sort of on the bad end of a stick here. His name is Hans Krebs. He was a Jewish biologist. He is a German Jewish biologist who ended up leaving Germany during World War II in order to pursue his research. He was, as you can imagine, being persecuted. So he left, and he decided to study how glucose is broken down in the body. So he did it using these pigeons. Now, unfortunately, your book, well, the previous edition of the book decided to call it the citric acid cycle because one of the primary products is citric acid. They decided not to name it after the Krebs cycle because there's this unfortunate misunderstanding where they thought that Hans Krebs was a Nazi, and they didn't want to name it after him because there was another Hans Krebs who was a German biologist during World War II, who was one of the war criminals in the concentration camps who experimented on Jewish people. So they decided, we're not going to name anything after a Nazi. And it turns out he wasn't a Nazi. He was one of the people fleeing the Nazis who ended up helping in the Allies in the war. So not cool for him. Anyways, here's the story. Hans Krebs and pigeons. In case you didn't know, when Krebs fled to England, there were lots and lots and lots of pigeons. If you've never seen a pigeon before, pigeons are effectively rats with wings. They eat anything. They flock everywhere. They defecate on anything, even you. If you guys have seen, like around here, when birds are migrating, if there's a bunch of birds sitting over top of your car, you move your car. Pigeons are everywhere. Nobody cares about them. The big thing to know about pigeons, though, is there are going to be old ladies who feed them. Be kind to old pigeon-feeding ladies, because they are inevitably not as insane as they look, and they can save you, as seen in Mary Poppins and Home Alone. Home Alone. So being nice to pigeon ladies is the same as being nice to, like, old blind kung fu masters. I mean, they may look like they're not much, but they're going to definitely save you later. Pigeons, rats with wings. So don't feel bad when I tell you that he decided to clip their wings. And by clip their wings, I don't mean just like a little clip. I mean off. He needed to culture their cells. And what he found was he extracted the muscle cells and studied how these muscles got their energy. As you can imagine, a pigeon has some powerful muscles to beat its wings. So he used inhibitors to stop this Krebs cycle at every single one of those little blue arrows to figure out what was being produced and what it was being broken down into. So he figured out how these carbohydrates were being broken down. And for that, he ended up winning the Nobel Prize. So this is what a Nobel Prize looks like. To me, it looks almost like chocolate. Like, I could totally see that as a chocolate coin. You know what I'm talking about? They're the terrible chocolate you get at Easter that nobody ever really wants, but you eat anyway because it's chocolate. So he won the Nobel Prize. Pigeons died. They died. But we did figure out this citric acid cycle, the Krebs cycle. As far as the Krebs cycle goes, it's all about counting the carbon. You start off, you've gone from pyruvate. How many carbon are in pyruvate? No, pyruvate up here. Remember, we go from 6 in glucose. We cut it in half to produce pyruvate. 3 carbon in pyruvate. Then you cut off 1 carbon dioxide from it. So now how many carbon are we down to? 2 carbon left, right? That's where we have our acetyl-CoA. 2 carbon. That 2 carbon acetyl-CoA goes into the Krebs cycle and gets attached to a 4 carbon molecule. That 4 carbon molecule gets cut and releases carbon dioxide. When you cut the bond, what else is released? Energy, and it'll be in the form of NADH. Then you're going to cut off another carbon dioxide, so now you're back down to 4 carbon. And you've released more energy in the form of NADH. You release some guanosine triphosphate, GTP, FADH2 because you cut another bond. You cut off another functional group with NADH. And we're back up to our 4-carbon that combined with 2 more acetyl-CoA to create our 6-carbon. And you have a cycle repeating. Where did I put that chalk? There it is. So here's the big overview. You go acetyl coenzyme A. It's going to be broken down in a cycle known as step 3, the Krebs cycle. That acetyl coenzyme A is going to hack off 1 carbon dioxide. And when that's broken, when you cut carbon dioxide, when you cut a bond, you release energy in the form of NADH. We go through, we're going to cut off another carbon dioxide. And as we do that, we release more NADH. So we've cut carbon dioxide. We cut carbon dioxide. Next, we break a functional group off to create a molecule called guanosine triphosphate, GTP. We break off another functional group to produce FADH2, another energy intermediary, and then another functional group to produce NADH. So for every 1 acetyl-CoA that goes in, you produce 1, 2, 3 NADH, 1 GTP, 1 FADH2. But we don't have just 1 going through. This cranks through 2 times because we have 2 acetyl coenzyme A. So for every 2 acetyl coenzyme A that go through, you produce 2 carbon dioxide, 6 NADH, 4 carbon dioxide, 2 GTP, 2 FADH2. So that's the Krebs cycle. You've produced a lot of energy intermediaries. How many, in this whole process, how many ATP are produced so far? What? 2 ATP. So we've produced, so far, 2 ATP. That's good, but enough energy to keep us going. How many FADH2? How about that? 2. We crank through twice. 2 FADH2. How many GTP? 2, because it cranks through twice. And now the super hard one that nobody gets right. How many NADH are produced? Okay, so where do you see 6? 8? Okay, where do you see 8? Okay, so we've got 2 NADH here, right? So that's 2. 2 NADH there, so that's 4, 5, 6, 7, 8, 9, 10. That one, sorry, that's my little arrows in the wrong place. I'll fix that. Is that better? All right, so we've produced 10 NADH. Crazy, right? I don't need you to know the numbers. I need you to understand we've produced a ton of energy intermediate, not much ATP. Each of these has excited electrons that can do work. So far, so good? Glycolysis, breakdown pyruvate, Krebs cycle, and what we've produced is a whole bunch of energy intermediaries. You got this GTP. All right, GTP and ATP are very, very similar to each other. As you can imagine, they both have TP in them. Triphosphate. They have the same basic function. The deal is, GTP and ATP are energetically similar, but they're used for different things. GTP is usually used to build proteins. I like to think of ATP as an American dollar and GTP as a Canadian dollar. They're worth about the same, but you don't use them to buy the same stuff. Does that make sense? They have about the same amount of energy, but they do slightly different things in the cell. So there we go, the citric acid cycle in all of its glory. I am not going to make you memorize this, but this gives you the idea of Let's see where we're going to come in at. We'll come in with acetyl coenzyme A, right up there. That acetyl coenzyme A is going to break down and release carbon dioxide and NADH. Then you've got citric acid. It's going to release some, and it's going to be broken and release NADH there, release more carbon dioxide, more NADH, some FADH2. Where'd the GTP go? Oh, here it is. They're saying ADP to ATP. It's actually GTP. You can read these things. Even though they look complex, they're just one step at a time. Does that make sense? I have faith in you guys. I know it looks hard and it looks rough, but you've got the basics behind it. Scientists really love to make what they're doing seem really difficult and really hard by throwing chemical formulas in there. But all I'm asking you to look at is the big picture. What you're doing is taking that acetyl coenzyme A and breaking it down to release energy intermediaries. That leads us into our final step, oxidative phosphorylation. Now remember, what organelle has all of this happened in? No, not the chloroplast. Mitochondria, cellular respiration. It's all happening in the mitochondria. The mitochondria is special. Why is the mitochondria special? It's like the chloroplast and the nucleus in this regard. Double membrane bound. So if you have your mitochondria, that is double membrane bound. Now this double membrane has all sorts of twists and turns in it. What do twists and turns do for the mitochondria? Increased surface area for reactions to occur. Because embedded in this membrane are all sorts of enzymes. Your citric acid cycle happens in here, your Krebs cycle. It produces all of those intermediaries. So the citric acid Krebs cycle happens in the inner membrane, inside. It's then going to pass all of those energy intermediaries to these energy acceptors, these electron acceptors in the membrane. This is going to seem really, really familiar to you, I hope. So you're going to take the NADHs, take those FADHs, they're little dump trucks filled with electrons that can do work. And you drop them off at the enzymes, at the proteins that are embedded in the membrane. And when you drop them off, they can do work. And the work that they do is push hydrogen from one side to the other. Then, the electron, having pushed hydrogen over, jumps to the next molecule that has slightly more electronegativity and pushes hydrogen across. And it jumps again, pushing hydrogen across. That's called the electron transport chain, where you're moving electrons from one point to the next to the next. Each time it jumps, it pushes hydrogen across. So what have you built up here? Lots and lots of hydrogen ions on one side of the gradient, not the other. The pH is going to go up. But what I'm more concerned about is the concentration of ions on one side or the other. So you created an electrochemical gradient. We saw that with photosynthesis. It's the same process of chemiosmosis. The final electron acceptor of the electron transport chain here is oxygen, which is converted into water. So you have lots of hydrogen on one side, not the other. How can that hydrogen get back in here, do you think? It doesn't break down. It's got to get in through a channel, right? What's the channel protein it uses? Same as in photosynthesis. Yes, ATP synthase. You have that ATP synthase over here. Hydrogen ions go through. As they go through, they spin it in a circle. That spinning creates a mechanical force that will take an ADP and a phosphate and crush them together. So ultimately what you're doing is you are taking the energy intermediaries from the citric acid cycle. They've got energetic electrons that can do work. You drop them off at these proteins. As that gets dropped off, it pushes hydrogen across. The electron then jumps to the next molecule that's more electronegative, pushing more hydrogen across. Then it jumps again, pushing more hydrogen across, until finally it reaches the final electron acceptor, oxygen, which is then converted from oxygen to water. That is why you need to breathe. You need to breathe. The whole reason you breathe is to get that oxygen. Without that, this process doesn't work. You don't get energy. So again, we're using that electron transport chain to move electrons and use the energy they have to get that hydrogen into the intermembrane space. They're passed from one molecule to the next, pushing that hydrogen till they reach oxygen. Oxygen is the final electron acceptor. When it accepts that when it accepts that electron, it becomes water. Waste product. Every time you're moving electrons through the chain, you're creating some water and pushing hydrogen in there. And again, what's it called when you push hydrogen in there? All of that hydrogen on one side. An electrochemical gradient, yes. It's a gradient. And you can use the power of that gradient to power ATP synthase. ATP synthase is going to be your big, efficient molecule for transforming that gradient into usable cellular energy, ATP. So the hydrogen ions stream through the ATP synthase, spinning it around like a water wheel, powering the creation of adenosine triphosphate. Now that ATP works, that ATP synthase works by having three different conformations, three different shapes. The first shape, conformation 1, you have the ADP and a phosphate. So let's say here. Here's our ADP, here's our phosphate, right? Step 1, first step. They bind to the ATP synthase, that's me. ADP, phosphate. Step 2, we're going to spin, and as we're spinning, it's going to take the phosphate and the ADP and put them together like that. Stage 3, that's released. Back now to stage 1 where I can accept more ADP and phosphate. So step 1, bind ADP and phosphate. Step 2, put them together. Step 3, release them. Now I'm back to the beginning so I can accept more. It spins and it spins and it spins, and each time it spins, it's making more ATP. This is extremely efficient. Questions on that? That's how ATP synthase works. In undergoing aerobic cellular respiration, you're able to produce 30 to 34 ATP per 1 molecule of glucose. That's how efficient it is. It's not just breaking glucose in half and releasing 2 ATP. You're producing 30 to 34 ATP. This gave us a major edge in becoming multicellular and taking over the world. We have 16 times more energy than those things that just break glucose in half and call it good. I do want you to understand for sure, for absolute crystal clear, that you don't just get energy from sugar. It comes from all sorts of sources. Carbohydrates, fats, proteins, anything you're eating, you can have go into this process. They just enter it at different points. They might enter during the Krebs cycle. They might enter somewhere else. But they're still broken down in the same manner. You need oxygen in order to process this stuff to get energy from your food efficiently. So here's the questions for the end of lecture questions. But I do want to point out, next time, we're talking about fermentation. Fermentation builds right into this process. So if you understand step 1, step 2, step 3, and step 4, fermentation is just a sidetrack. You're going from pyruvate There's no oxygen present in fermentation, and you're producing waste product. In the process, you convert NAD plus into NADH. That's going to be the whole of next lecture. So if you get this, you've got the hard stuff. There's some other stuff I wanted to point out very briefly. Right. I do recommend going through, drawing this, working with people who don't understand it, do it over and over and over again. In your lab, you're going to be asked to do flow charts. That's what that is. These flow charts, if you haven't invested in a whiteboard, get one and draw this out again and again and again.